Yeah, that's a nice question because I was not heading for neuromuscular actually. I was working as a regular neurologist in a regional hospital in Finland, and just happened to see peculiar patients with peculiar disease. And over the years I understood that the muscle disease they have has not been described anywhere in the world. So it was a challenge for me to step into that from nowhere. Yeah, so continuing on my previous statement, these patients having a new type of muscle disease, they challenged me, and I had to, of course, start to try to first describe the disease, what it looked like, and then collected large families. It was primarily dominantly inherited disease, late onset distal myopathy. And at the time, we are talking about 40 and 45 years ago. At the time, there were no molecular genetic possibilities to have a clarification on the genetic level. But I collected these families because I knew it was coming. So in the mid-80s and late 80s, we then started to collect DNA samples of these families and do linkage studies. And that is the way it started. Of course, with the technique at the time, everything took very, very long times. And we continued with the linkage studies almost 10 years and did not get anywhere. So what to do, to leave the whole project or not? I had to readjust something, and the question and the problem was actually that there were two phenotypes in the families, in some of those families. Most had just the dominant late onset distal myopathy, which I then reported as with the name of tibial muscular dystrophy. But in some of the families there was another phenotype. In a few cases, early onset limb girdles. And because in one family both parents had the distal, we then of course thought that these severe cases were homozygous for a genetic defect. So that was our concept, but it was not successful. So after almost 10 years of struggling, I decided to change the concept and collect only the families with the single late onset distal myopathy phenotype. And then we had the linkage within 1 year. Okay. And that was the starting for getting on to find the gene. Yeah, so I have not given the name. Okay. That was a sort of invention by OMIM to do so, and I have no responsibility for that. But of course, I'm not very unhappy with it either. So. Oh yes. With titin defects. Yeah. So currently we know of 10 different disease manifestations, and there are some more easy parts of it. So if you have a biallelic truncating mutation in titin, you usually get a severe congenital titinopathy. There are differences depending on where the locations are, but still. And then we have my distal titinopathy, the tibial muscular dystrophy with the founder mutation, Finnish founder mutation in the last exon. And that was, of course, then the starting point to see whether that disease phenotype existed in other countries without our Finnish founder mutation, and that was the case. So we reported families from France, from Italy, and other countries with other mutations in that specific last exon. And for those who are not that familiar with genes and genetics, just to tell you, thytin is the biggest gene of all genes we have. And the protein it encodes is the largest protein in nature, not only in the human being. So it was a real challenge, and after we had the linkage, of course, to find the solution for on the genetic level. And at that time, we did not have any next generation sequencing. So thytin is compiled of 364 exons. And with the techniques at the time when we started to sequence thytin 25 years ago, after having the linkage, you had to make primers for all parts individually and sequence one part at a time, and we started with exon 1 and went on for 2 years until we came to exon 364 and found the mutation responsible, the Finnish founder mutation. Yes, we are underway. We have mouse models for the Finnish founder mutation in Paris, and we are doing sort of tissue modeling of by cells, and can introduce different mutations in that model system, and it's the aim is to go then for sort of large-scale approaches to see if any drugs can make a difference. There is a peculiarity with that last exon mutation system, because on the protein level it seems to induce pathological cleavage of the protein. So a larger part of the C-terminus of the protein is cut off. For some reason, the mutation induces that pathological cleavage, and that is now under study to see which drugs can prevent that. So it's an easy explanation, because after I collected all types of distal myopathies, and after having clarifying the distal thytinopathy, the thytin defects, we were left with a large bunch of unsolved distal myopathy patients. So I can't leave that. I have to go for clarification also of the unsolved cases. And we very early, we went into the next generation sequencing techniques very quickly. So 10 years ago for all clinical cases, and with a gene panel. But on that gene panel we did not put only known genes, but also genes that were highly sort of good candidates for making muscle diseases if defect, and that was very successful. So we actually identified half a dozen of new diseases just that way. The expectations are high. And of course, this is a new field, which means that all sort of tries to do something will not be successful. And that is just part of science. You have to have a hypothesis, what could be of benefit, and then make it coming through and see if it works or not. And that is now the current situation. We will have pitfalls, we will have some successes, and it will take a long time until we have the gene editing tools in place to fix it all. So you just can have a look at how this World Muscle Society Congress has grown over the years. And you see, I'm so happy to see the growing interest of neuromuscular, because in the past in neurology, neuromuscular was also always on the backstage. More important was multiple sclerosis, epilepsy, and stroke, and Parkinson's, and ALS, and all other things. And neuromuscular was not appreciated. But that is changing. And part of it is, of course, in the advance of the genetics, where neuromuscular has been in the forefront. And also in the now in the stage of early therapies. So from at the first WMS meeting in London, 180 participants. We are now over 1,000. So I'm happy. Okay, so that's an interesting question. I love to work in the forest, taking down trees at my summer house, making firewood. I do not need to go to the gym if I do that. So that's among other things, and go fishing our own fish for the whole year and things like that, yes, besides science. Yeah, I think I can easily answer that by telling that after graduating or having my specialty as a neurologist, I went back to my hometown. Okay. And started to renew an old house next to my home house. So that maybe tells a bit about affections.